My name is Mauro Zappaterra, and I'm a physician. I did my MD, PhD at Harvard Medical School. Now I have a private practice in physical medicine and rehabilitation in Pasadena, California. I'm also working with the Los Angeles VA healthcare system. And I've been trained in polarity therapy, Reiki, and craniosacral therapy as well. So I try to integrate all the trainings that I've had and try to care for patients. And physical medicine rehabilitation is a specialty that essentially tries to optimize human functioning in whatever you may have. So if it's a painful elbow, we try to either decrease the pain or help you with movement of the elbow. If it's a spinal cord injury, we try to help you with any of the what we call activities of daily living. So that's, washing your face, brushing your teeth, going to the bathroom, dressing yourself, stuff like that. How can you optimize your function to live your life and increase your quality of life? So sometimes we need to use medications. Sometimes we might need to do surgeries. I don't do the surgeries myself, but I usually, I'll refer to somebody. Sometimes they might need some therapy, like physical therapy or occupational therapy. Sometimes they might need something more energetic, like Reiki or craniosacral therapy. Typically, if I feel like they need something more energetic, if I feel like there's sort of some existential suffering that's occurring that, let's say, unfortunately can't be dealt with in the amount of time that I have to deal with them, I'll actually refer them to other people. And I refer them to my wife, usually, who does craniosacral therapy. So the CSF stands for cerebrospinal fluid. And I did my entire PhD dissertation on this fluid at Harvard Medical School. And essentially the way that it came up was that, believe it or not, I thought that I wanted to do research on cancer. And I was in a cancer lab, and I was not so happy, let's say, in the cancer lab. And my wife says, she comes to me and says, I don't think you're very happy doing what you're doing. I think you need to sort of shift. Let's take a break. And so we took a break and went to Santa Fe together, and she wanted to study polarity therapy, which is a holistic healthcare system. And in one of the classes, we were learning some craniosacral techniques. And the teacher said, you're working here with the cerebrospinal fluid. The cerebrospinal fluid is one molecule away from seawater. And I said, well, that's interesting. Like, seawater is an interesting, compound. We have this fluid inside of us that's one molecule away from seawater. So that night I go home and I start kind of reading up on the cerebrospinal fluid, and I realized that there was very little known about this fluid. Very little known about this fluid. It was thought of sort of providing a couple nutrients and sort of buoyancy and protection to the central nervous system. So as we go throughout the training of our polarity, I'm starting to think about going back to Harvard to finish my PhD, or to actually to start it. And so I left all the cancer stuff behind and started a lab that was able to actually investigate the question, what is the cerebrospinal fluid? And so in all of us, we have 150 milliliters of cerebrospinal fluid in our brain, inside of our brain, covering the outside of our brain, within the central canal of our spinal cord and around our spinal cord. 150 milliliters is equivalent of almost like half a can of Sprite or Coca-Cola. Every day it turns over about 3 to 4 times. So that means you make 500 milliliters, which is half a liter of cerebrospinal fluid a day. So we're all making this fluid, and it's bathing our entire brain. It's bathing all the insides of our brain, the outsides of our brain. The central canal, the spinal cord actually has a central canal that is bathed completely with CSF all the way down the spinal cord. And the CSF goes, and it actually goes all the way outside the spinal cord, all the way down to the sacrum. So what we want to do is essentially ask the question, what does the cerebrospinal fluid do? And so we did some experiments. We were the first to analyze very descriptively human embryonic cerebrospinal fluid. And we compared it to rat embryonic cerebrospinal fluid. And the reason why we did that was because I looked at some sections of some embryos. And I saw that in these sections, the cerebrospinal fluid space, the space in these sections was ginormous. It was huge. But the developing brain was this little tiny sliver. And inside was this huge space. And I actually turned to my colleague and I said, there seems like there's something missing here. And he goes, well, no, that's actually, that's where the choroid plexus is, which makes the cerebrospinal fluid. And I said, wow, all that is fluid? Well, what does that do? So first we wanted to analyze what actually was in the fluid. And then we wanted to see what does a fluid do from a developmental perspective. And so we kind of did some experiments where we mixed and matched certain sort of developmentally staged brains with different developmentally staged CSFs to see what does the CSF actually provide to the developing brain. And interestingly, what we found out was, well, first of all, just anatomically, every single neural stem cell in development, which are the cells that develop into neurons to create the entire central nervous system. Every single one of those stem cells at one point in development makes contact with the CSF or is contacting the CSF. So early on in development, you're actually making contact with these cells are making contact with the CSF. There's various cues and growth factors and molecules that are actually released into the CSF that instruct the developing brain on proliferation. How much do I actually need to expand, for instance? Differentiation. When should a cell that's a neural stem cell leave the sort of niche that it's in and go out and become a neuron so it can function and can have a more specialized function. And so we essentially categorized the embryonic cerebrospinal fluid in humans, as well as we did an extensive analysis in rats, looked at all the proteins, looked at how they changed and varied with time, and what were some of the functions of that. And what we published was essentially that the cerebrospinal fluid creates a niche for stem cells and provides information based on the molecules that are actually in the cerebrospinal fluid, provides information to the brain for proliferation or differentiation. And so that was kind of our major, finding with my PhD work. So in back of that then was, all this kind of interesting training from polarity therapy and craniosacral therapy of this fluid actually having sort of a spiritual component. And during my PhD thesis, one of the books I read was actually the Tibetan Book of Living and Dying by Sogyal Rinpoche. And he actually describes this practice. It's a POA practice, and it's done. It's a Buddhist meditation practice where essentially it's done at the time of death, and it's transferring one's consciousness to the unified Buddha consciousness. And there's a couple sort of indicators that you are successful in the practice. One of which is a tingling at the top of the head. Another one is headache. One is a small little hole appears on the top of the head where a blade of grass can be put into it. The other one is a softening of the fontanelle, which is where all the bones of the skull come together. When we're babies, there's sort of a space there, and you can actually push on it, and that's actually you're pushing on the cerebrospinal fluid. And so when somebody dies, that space actually softens, and you might have a little bit of clear fluid that actually comes out from the head. And so I said, wow, is that cerebrospinal fluid? If this is a successful practice of transferring your consciousness to the Buddha consciousness, and one of the indicators of success in that practice is a clear fluid that emanates from the top of the head, could that actually be cerebrospinal fluid? Because that's the only clear fluid that's present at that. So we actually tried a little bit to get some fluid from practitioners who did this practice, and we weren't able to do that. But those thoughts were kind of in the back of of at least my awareness and consciousness when I was doing this work. Combined with sort of my craniosacral training, it became a very powerful fluid. And I started doing, with the guidance from my wife and some of my teachers and stuff like that, some meditations on the cerebrospinal fluid. So for instance, just anatomically, we have a third ventricle, which is essentially like if you go mid-ear length, straight line, and then midline, right in between your brow, there's a fluid-filled space. It's in the midline. It's called the third ventricle. Just in front of the third ventricle is the pituitary gland. Just in back of the third ventricle is the pineal gland. Okay, both those glands are contacting the third ventricle. This third ventricle then opens up. It goes up into the lateral ventricles, which are on either side of the brain, and it goes down all the way the central canal of your spinal cord. Okay, in Kundalini yoga practices and traditions, they describe a hollow canal in the center of the spinal cord where the Kundalini rises and essentially brings the awakening of the Kundalini, or this primal energy, to the third chakra, where then it goes up to the crown chakra and an awakening occurs. The caduceus, for instance, is the midline. So the staff of the caduceus is sort of like a midline. And the two sides of the caduceus are depictions of two of the channels from this yogic practice, which would be the Pingala on one side, the right side, Ida on the other side, the left side, and they cross. And at each crossing with the Sushumna, they make a chakra. The meeting of the heads, which are the heads of the serpent, come together at the midline, and they come right at the third eye. In my belief, the heads of the snake represent one head of the snake represents the pineal gland, the other head of the snake represents the pituitary gland, and the center is the Sushumna, which is the cerebrospinal fluid. When they come and meet together, it's sort of this meeting of the three energies right in the center here. The third eye, sort of the eye of the soul. This place has been called the Crystal Palace. It's been called the Cave of Brahma. And so from that space then, it is my belief that, and some other people's belief as well, that there may be a physical manifestation of the sensation of I am, our beingness, at that space when these energies come and combine, that we have this sort of sensation of beingness. And the reason I say that is because the I am is a fluid. The I am can transmit, the cerebrospinal fluid is a fluid. The fluid is known to transmit light. It can transmit vibration. It can transmit flow, so flow from the fluid, and it can even translate molecules. So if there's sort of an experience of a unified experience that you're having, the cerebrospinal fluid, bathing all the areas of the brain that it bathes, has no synapses that it actually has to go through. If a certain message, let's say, is dispersed throughout the fluid, it can go to all the major control centers of the brain. It can be done totally non-synaptically, and it can have this whole synchronized effect on the brain because it's bathing the entire central nervous system. Let's say the guiding light from this talk for this talk really came from talking to you and Maurizio, who stated that, I was talking to you about the fluid and you said, well, you know what Nisargadatta said about this fluid? And I said, no, I don't. And he said, well, he said the fluids come together and the sense I am appears. And I think instantaneously we kind of looked at each other and said, maybe that's the cerebrospinal fluid. And so that's exactly what, what I'm talking about is sort of when there's a sense of I amness, when this sense arises, when that inner sense of beingness arises, it's my experience that it's sort of I sense it in my third ventricle or my third eye, and then from there it gets into this expansion, sort of transmitted through the fluid to my entire central nervous system, of which it's just a complete holistic feeling of my sense, or the sense of beingness. And so that's in essence the talk that I give, branching it from, here's the anatomy. As simple as that might be, we have a fluid-filled space right in the middle of our brains that has this fluid. We have a canal that goes down the center of our spinal cord. And when you look up, sushumna, it sort of describes a hollow canal down the center of the spinal cord. It can transmit information. If you look at flower essences, or the work of Masaru Emoto, who looked at, how do fluids store and transmit energy. So here is a fluid that can, store, even if we're just looking at a molecule, it's storing the molecule and it's transmitting that molecule to another source, or to another space, being transported via the CSF. It can transmit light, it can transmit vibrations. And in my talk, I use sort of melatonin as an example, because melatonin is synthesized by the pineal gland. And it's synthesized by the pineal gland. It's mostly synthesized, it starts being synthesized when the sun goes down, and it helps us sort of induce a state of sleep. So that's already an altered state of consciousness when you actually fall asleep. So here's this gland, it starts secreting melatonin. There's really good research out there that suggests that the melatonin actually goes into the CSF, and the CSF then distributes it to the brain so that we slowly fall asleep. Interestingly, DMT, which is dimethyltryptamine, is also synthesized in the pineal gland. Rick Strassman found it in 2013 and published a paper. And DMT is used in many shamanic practices to sort of induce altered states of consciousness, as well as it's believed to be hypothesized to be released endogenously during birth, death, and during lucid dreaming. So, if there's these molecules in the CSF, like melatonin, that are being, released into the CSF and then distributed to have this sort of global experience of an altered state of consciousness, like sleep, could then the CSF store and transmit energy from, let's say, a source energy, transmit that energy to our beingness, to our sense, to ourselves, and allow us to experience our sense of self, that pure beingness within us, and be aware of that. So that is sort of the essence of, kind of the views of the CSF that I wanted to present to SANS. So the structure of the CSF can change based on nutrition. I feel like it can change based on exercise. It can change based on, traumas. So people are, they're always asking me I have a concussion, what can I do? We know that the CSF is changed if there's a stroke or a bleed. Sometimes the blood might actually get into the CSF. But even just in your sleep, for instance, just as you go throughout the day, even if you look at something like melatonin, for instance, it's like this, let's say you wake up, okay? It's a nice, and then if you collect CSF, and then around 7 o'clock or 8 o'clock at night, it starts to increase in your CSF. It increases for about 6 hours, and then it starts to decrease in your CSF. So that's just one molecule that we've looked at closely. In development, what's interesting when we looked at in development, because we were able to take, let's say, like embryonic day 7 and embryonic day 10 and embryonic day 15 and 17 and, newborn and adult rats, for instance, and see how does it change. Every single protein we looked at was different. It was like, we had some proteins that came up early and then went down. We had some proteins that actually came up late, were mostly present in the adult. We had some proteins, for instance, that stayed very constant. And so, we're very interested by obviously the ones that are changed. It's like, wow, we saw just like a little blip for 2 days. What's happening in the brain during that little blip. And that's how we kind of went back and said, is this a major part of neurogenesis, or where a lot of the neurons are being made? And so what we found was, specifically, there was one molecule. Again, we're always kind of breaking it down into molecules, but we found one molecule that changed. It had a little blip, and we looked specifically at that time point, and we saw that that was a major time point for the majority of the neurons to be developed. And so, so it's changing all the time based on, what you're doing. The best thing I can say is, you try to live healthfully, and so you have a healthy CSF, you exercise, eat good food, because all that's going to actually change what what's present in the CSF. It's in constant flux. You turn it over 3 to 4 times a day, so you're constantly making new fluid. And if you look at the sides, if you look at the CSF comes and it contacts the ventricles. If you look at the cells of the ventricle, for instance, these look like little hair-like projections. They have hair-like projections on them, which are called cilia. And these cilia can actually beat too. So these cilia can kind of create a movement in the CSF. It's like, and there's experiments, for instance, where they've taken, a rat, they opened up, a rat ventricle, and they put a little dye or a little bead on one side, and they wait, a few minutes, and the dye is migrated all the way to the other side. Or, the bead has actually moved because these cilia are kind of beaten. So just kind of imagine, here's this fluid-filled space. Here are these fluid-filled spaces in our brain that is moving, is constantly in movement, right? That transmits. It can transmit light. It can transmit movement and vibration. It can transmit molecules and growth factors and ions and hormones. And it's constantly moving. It's constantly in flux. It goes all the way from both sides of your brain to the third ventricle, which is right in the middle, contacting both the pituitary gland in front and the pineal gland in back, going all the way down your spinal cord in the middle. So in the middle of your spinal cord, on the outside of your spinal cord, all the way down to your sacrum. So if you can kind of imagine this fluid, bathing this whole area, and then once I do that, I sort of connect into the fluid nature of my being, and just connect in there. And the thing that I'm interested in is when I sort of bring up the cerebrospinal fluid, or when I talk about it, and I have people connect in with their own cerebrospinal fluid. What is your experience of it? How do you experience the fluid nature of yourself once you kind of get a sense that, wow, there's this fluid in my, there's fluid in my brain. It goes all the way down to my sacrum. Does it have any qualities for you? And so I'm very interested in your experience, because I have my experience, but I'm very interested in everybody's experience. Once they kind of connect in with the fluid, how does that help them? And is there some sort of relationship between the fluid field of the cerebrospinal fluid and the heart? And so what is that relationship? And that's something that I'm sort of personally investigating myself. Even with our PhD work, for instance, I was very fortunate to be in Chris Walsh's lab, who, he had funding to do these projects, because it took a lot of work to actually do these projects. And who would allow themselves to have a lumbar puncture done while they're in a deep state of meditation, or something like that? Some of the projects that, for instance, we're looking at is, even Rick Strassman, for instance, trying to find DMT in the CSF. Very difficult to find one molecule in the CSF, because there's so many other molecules that are larger and there's so much more quantity. So when you're looking for one, it makes it very challenging. And as better techniques get designed to actually look for those little small molecules that actually just come at moments, just a tiny little moment. How do we find that one? So, something, for instance, like, even studying the process of dying. We really have very little information, but I know that now people are starting to look at, well, what is the neural activity in the brain that's happening during death and then at death, and when do we say death? Could we actually go and, unfortunately, we do these studies on animals, but can we go during a moment of death and take the CSF, and see, is DMT actually, is there a higher concentration at that time? And those are things where essentially you need funding to do those sorts of studies. But we tried looking, like I was describing the POA practice in this Buddhist, we tried finding anybody that would, but just kind of imagine, trying to set up a scenario where it's like, well, was that a successful POA? Oh yeah, here's the fluid. Can we go in, collect a little bit of this fluid just to send it to this laboratory so that they can analyze it? But that's what we would love, from my perspective, I'd love to know, what is that clear fluid at the top of the head if a POA practice is actually successful? Does it have any constituents that are similar to the CSF, or is it more like salivary, fluid or something like that? But it's also experiential, so how are you, when you meditate on your third eye, or when you meditate on your third ventricle, what is your experience? Where do you feel the energy? Do you feel energy? Do you feel anything building? Do you feel any movement or vibrations occurring? Do you feel any subtle movements or subtle energies in your sacrum? Do you feel an expansion? Do you feel a contraction? And those are kinds of the things that, it's like kind of building up personal stories from people and saying, well, this is what people's experience are of it, and going from there. So they are trying to look for receptors for, DMT and stuff like that. One of the reasons I believe, I think you can inhale it, you can snort it. One of the reasons I think some people actually prefer, regardless, it's going to get to your brain. And, I don't know how the distribution is when you actually exogenously deliver it, but it's going to get to your brain somehow, and that's how the effect is. In the inhalation process, for snorting it, there are actually, the cribriform plate is this bone that sort of comes out, and coming out of the cribriform plate is actually your olfactory nerves. And so it's the one place where you can actually penetrate the blood-brain barrier. And so people are actually trying to get, therapeutic molecules into the CSF using intranasal intranasal approaches. So I'm using an intranasal approach to actually get to my brain faster via the olfactory nerves that are open, that there's actually a sort of an opening in the blood-brain barrier at that area, and you can get stuff into the CSF. And so what they're looking at is, how do we encapsulate these possible, therapeutic medicines, so that because it has to travel through, essentially up the nose, through the brain material, through the brain tissue into the CSF, because that's really the only way we know how to get into it. Or, can we take something like eat it, and it goes through the blood, and then it gets into the cerebrospinal fluid. So I'm not quite sure on that, but the other way is obviously, putting a shunt. They call it a ventricular shunt, and going right into the ventricle and change. We can do that in animals. In terms of changing the properties of the CSF and seeing what this molecule does or what this molecule does. In terms of whether or not the cerebrospinal fluid is one molecule away from seawater, it turns out that it's not, because we have a lot of other proteins and hormones and growth factors that are actually present in the cerebrospinal fluid that aren't present in seawater. However, and this is fascinating to me, evolutionarily, when people have looked at the evolution of what they call the CSF contacting neurons, so these are the neurons that are contacting the CSF. Okay, when they look at something like the starfish, for instance, or an eel, what they find is that the same neurons that contact the seawater in a starfish are evolutionarily the neurons that are contacting the CSF in humans. So therefore, could you see the CSF as sort of our internal ocean that evolutionarily developed, that we are monitoring the ocean? And so that's the ocean within. So that's the fluid. They're actually calling them the CSF contacting neurons are contacting the seawater in a starfish is contacting the CSF in us. So we have this ocean. We essentially have the ocean within us. Every drop is the same. So that to me is the beauty of this fluid, continuously sort of investigating it and picking it apart and sort of looking at it closer and closer and farther. So the CSF, where it comes from, is also a very interesting topic. Initially, when you're an embryo, okay, you're sort of a flat sheet of cells, okay? I'm going to say on top of you, but on top of you is actually what's called the amniotic fluid, okay? So already, even before there's any left or right or anything like that, you're a sheet of cells, and on top of you is the amniotic fluid. As you start to develop and there starts to be an invagination or an involution of the cells, what happens is that there's sort of this sort of a flat sheet, and it dives down and then it comes up and it folds in on itself. Okay? So this diving down and folding in on itself process, what it does is, if you can imagine fluid here and then diving down and folding in, it folds and it kind of makes a tube. That tube becomes your spinal cord. So essentially it's picking up all the amniotic fluid, right? And then creating, once it's sort of separated from the amniotic fluid, then we call it CSF because it's in the middle now of the tube. And as it sort of develops, then even the ventricles come, or the two sides of the brain come on top and come together. So initially the CSF is actually amniotic fluid. Then, as development starts to occur, we start to get these specialized cells called choroid plexus. And choroid plexus are very vascular cells. They have a really intense blood supply. And so the choroid plexus is the main sort of supplier of cerebrospinal fluid throughout our entire life. But I believe as people start to investigate more about the fluid, the more that we're going to see that it's not only the choroid plexus, but it's actually coming from even the cells, so the brain tissue. So the brain tissue itself releases fluid into the cerebrospinal fluid, as well as some interstitial, the fluid within the actual cells, some interstitial fluid that actually comes and gives rise to the CSF. So right now, the two main areas where the cerebrospinal fluid is coming from is the choroid plexus and the interstitial fluid, or the fluid between the cells of the brain, and that's where it's being produced.